Okay, I think we'll go ahead and get started. Thank you all for joining us this evening. I'm Elizabeth Harper. I'm the Executive Director here at the Lake Sunapee Protective Association. Our mission here is to preserve and enhance the environmental integrity of the Lake Sunapee watershed. And we do that through education, research, and collaborative action. Thank you to all of you who are members and support our work through your membership donations. That allows us to do the work that we do here at LSPA. And we appreciate seeing a lot of our members on the list of those of you who are joining us tonight. Our speaker tonight is Dr. Celia Chen. And before she gets started on her presentation, just a few reminders. We'll keep participants muted for the duration of the presentation. If you do have questions, feel free to enter those into the chat. Kathleen and Nancy will be monitoring the chat. And if there are questions that are timely, relate to what Celia is talking to at the moment, we'll go ahead and have those questions asked as we go. And then we'll have time for questions at the end as well. We are recording the presentation and we'll share that link with you in a follow-up email in a couple of days, along with some links to additional information. If your camera's on, everyone can see you, and you can turn your camera off if you prefer. It's nice to see your faces sometimes as well. And please, if you'd like to, certainly turn your cameras on at the end so we can have a little bit of a discussion and questions at the end. So if there aren't any questions before we get started, then I'll go ahead and introduce our speaker tonight. Dr. Celia Chen is a research professor in the Department of Biological Sciences at Dartmouth College. She's also on LSPA's Scientific Advisory Committee. And tonight she's going to speak to us about PFAS and their bioaccumulation and fate in marine food webs. So thank you so much, Celia, for joining us tonight.
You're welcome. Thanks all of you for attending tonight. It's nice outside here, so maybe you would have rather been outside, but glad to have you here. And I'm going to talk to you tonight about research that we've been doing at Dartmouth on PFAS, and it seems to be the contaminant of the hour, I would say, these days. You might have heard a lot about it on the news because the US EPA just released their final decision on the regulation for the maximum concentration level for PFAS in drinking water. That has big implications for everyone. I won't talk so much about that, but needless to say, PFAS has been in the news a lot. And so as Elizabeth said, I'm a research professor at Dartmouth, and I have been studying contaminants for all of my career. We spent a lot of years studying mercury, but in recent years, mercury has really gotten to be, it's very well studied, but we've really moved into the phase of sort of global controls, international treaties. We're working on mercury at that level these days. PFAS is a much less understood contaminant, and it's very complex. So tonight I'm going to talk to you about some of the overarching questions that we ask. Basically, things like the chemical, physical, and ecological factors that determine bioaccumulation and trophic transfer of PFAS in aquatic food webs. So we're food web scientists, and we like to know, how does basically these contaminants get into the organisms that transfer the contaminant up to some of the species that humans consume. And we've worked in lakes and estuaries and in coastal waters. But tonight I'm going to tell you more about marine PFAS research that we've been doing. We are now working in freshwater as well, and I'll tell you a tiny bit about that. But tonight I'm going to cover some things, a study that we did basically, which is a review of PFAS compounds in shellfish, in demersal fish in the Gulf of Maine, shellfish in the Great Bay, another study that we're working on and writing up, and also a recent study that just came out a couple weeks ago on New Hampshire seafood consumption, and a market basket study showing our sort of New Hampshire seafood consumer exposure to PFAS. So before I launch into our research, I'm going to give you some background on PFAS. It's something that a lot of people either never heard about, or if they heard about it, they're not sure what it really is. So PFAS are chemicals that are basically produced by humans and put into the environment through all kinds of uses that we have found for these incredible class of chemicals. There are over 12,000 PFAS. PFAS stand for per and polyfluorinated alkyl substances. Better and easier to say PFAS. And they are products that were manufactured, produced, designed by companies like 3M and DuPont, and they have incredibly desirable qualities because they are heat resistant, oil resistant, stain resistant, and water resistant. And so they are in a lot of our products, including, you probably remember using a lot of Teflon pans. That was a real innovation at the time. PFAS is in Gore-Tex. Gore-Tex actually is expanded Teflon, and Teflon and Gore-Tex are both, really PFAS. And because of this amazing set of properties, they're in everything, in our clothing, in our furniture, adhesives, food packaging, heat-resistant nonstick cooking surfaces, and things like insulation of electrical wire, coatings on solar panels, and also in aqueous firefighting foams, which are shown in the two photographs on the left. And those have ended up contaminating a lot of water systems, drinking water systems. So why do we even worry about PFAS? Well, we know now through a lot of studies that that have been done and are emerging that there are a lot of effects on human health and likely wildlife health, although that's less studied. And there are a number of different ways in which PFAS affect our health. So on this figure, which came out in a paper in 2020, it shows solid lines for the kinds of human health effects that are really much more well-known, have high certainty associations, whereas the ones that have dotted lines are less, are low certainty associations. So, for example, thyroid disease, and I don't know if you can see my little arrow here. Thyroid disease, increased cholesterol levels, liver damage, kidney cancer, testicular cancer are all fairly well-established effects of PFAS exposure. You might have heard that, for instance, a lot of the firefighters who have used these firefighting foams, which are made of PFAS, there has been a high incidence of testicular cancer in firefighters, for example. We also know that there is reduced response to vaccines of the growing fetus, lower birth weights, as well as delayed mammary gland development in the growing fetus. In fact, the reduced response to vaccines was the endpoint that they used to establish the new maximum concentration level, the EPA MCL that just came out. But there are other ones that are very much in study, like the association of, fetus and children with early puberty onset, obesity, miscarriage risk, low sperm count, things like associated with pregnancy. So there's a lot of concern around the exposures of pregnant women and children. Just like for many of our contaminants, they are the most vulnerable populations. So what are PFAS? Well, some people have described PFAS like a family, and a family tree kind of shown here is how we think about PFAS. So you have the overall class of over 12,000 compounds at the top. We have non-polymers and we have polymers. We also, within non-polymers, have the perfluoroalkyl substances, and we have the polyfluoroalkyl substances. And really what we mostly, our lab anyway, focuses on are this group, the perfluoroalkyl substances, particularly perfluoroalkyl acids, the PFAS. And in this PFAS, we're going to talk about what are called perfluoroalkyl carboxylates, the PFCAs, and the perfluoroalkyl sulfonates. And I know this is getting to be a alphabet soup of lots of chemical names, but I'd like to just explain it a little bit in the best way possible so that when we're talking about these different compounds, you'll understand that they all behave differently. And so anyway, so I like these set of figures here because they help me think about them. And believe me, I am not a chemist. I'm an aquatic ecologist, and I have done the deep dive into the chemistry of these compounds, and I'm still very low on the learning curve. So I realize that I am still learning about these compounds, but just so that we are kind of on the same page. So these are the PFAS. And as I said before, there are the sulfonates and we have the carboxylates. And the difference between the sulfonates and the carboxylates is the sulfonates have a sulfur head. It's just it is a chemical construct that's attached to a chain of carbons that are attached to fluorines. And these PFAS are characterized by the fact that, if you can look on the right here, there are a bunch of carbons that are all attached to fluorines. And this carbon-fluorine bond is very, very stable, very tight. And so the reason that PFAS are called forever chemicals is it's really hard to break these bonds. And so they stay in this configuration in the environment for a long, long time. There's a lot of research now attempting to determine how best to destroy these organic compounds, but takes a lot of energy, and there's also not really successful techniques that are able to be used on the large scale. So, as I said, we have the sulfonates and the carboxylates. And if you look at this, these two compounds each have actually 8 carbons with fluorines attached. And the 8 carbon with fluorines attached with the sulfur head is called PFOS, PFOS. And the other one that has the carbon dioxide head is called PFOA. So you'll often hear people talk about PFOS and PFOA. They are the sulfonate with 8 carbons and then the carboxylate with 8 carbons. And this will come up again. I like the table below because it really shows you this alphabet soup of the number of carbons in the carbon chain. And on the top here, so you'll see the number 8 has PFOA and PFOS, as I just described above. But the other ones with 4 carbon chains are PFBA for the carboxylates and the PFBS. It's all an alphabet soup. These acronyms stand for very long chemical names, and I don't even ever remember what they are. So I go with the acronyms. And you can see, so the 5 carbon chain have PFPEA and PFPES. And then it goes up even beyond 12 carbon chains up to that are being measured currently, up to 15, 18 carbons. And so that's sort of the families and the organization of PFAS. And these ones that I've just highlighted with red squares, boxes, are the ones that are actually covered under this new EPA MCL rule. And so for drinking water. And so again, you'll probably see those PFHXS, HXS, which is a sulfonate, the 2 PFOAs and the 8 carbon, and then we have the 9 carbon carboxylate, which is PFNA. These are the ones that are going to be regulated going forward in drinking water at very low concentrations, including one more that's not on this table called Gen X. And we're not going to talk that much about it, but Gen X was a shorter chain PFAS that was manufactured to try to get shorter chain PFAS, thinking that they would actually be less persistent in the environment and less harmful to human health. But as it's turned out, that actually wasn't true. So Gen X is also regulated. I like this, and it might be a little too much, but so I said there's like short chains, and they're all the ones at the top here. And short chain carbon PFAS are more soluble in water. They have a higher water solubility, which means that they like to be in water. We also have, and so because they like to stay in water, they're not very common in fish, for instance. And we're going to eventually talk about fish in a moment. But the longer chain, which are like up, as I was saying, beyond 12, are uncommon in water. They tend to stay in the sediments in water bodies, and they tend to be much more common in fish. Oh, and I should say this set of figures is from my colleague, Tom Danielson at the Maine Department of Environmental Protection. I really liked the way he explained it. I saw him at a talk, and so he lent me his slides. And so, but these ones that are in the middle, I guess kind of like just right, they're medium size. They are found in water and in fish, and they are PFOA, as I just mentioned before, the eight carbon chain, PFOS, and PFNA, which is a C9, a 9 carbon. And these are the ones, again, that are the ones that are now regulated by the US EPA drinking water regulation. So how are we exposed to these? Well, as it turns out, since they are so ubiquitous in everything that we buy, use, and find in the environment, they are actually found in a lot of places. So at the top, you can see, I've already told you, they're found in a lot of consumer products. They're also emitted and discharged from industries like manufacturing, metal plating, semiconductor production, textiles. Lots of different industries have emitted them into the atmosphere and discharged them into waterways. We also have those AFFF foams that I showed you the picture of earlier. And because those have been used in fire training at the Department of Defense sites, of the Army, the Navy, they are also in the environment in many places now that are considered Superfund sites, because they have been used for practice and also putting out fires. And then what has happened is that they have then seeped into the groundwater, and they've contaminated a lot of drinking water systems. And you might have heard about a lot of them in New Hampshire, including the former Pease Air Force Base. There's also several manufacturing sites that are also contaminated with PFAS in New Hampshire. And the other place that you actually might not have realized is that because of all these consumer products and all of our wastewater going to sewage treatment plants that separate water effluent from biosolids, they have ended up being discharged from sewage treatment plants and also concentrated in the biosolids. And some of you may know that biosolids have been used throughout the country as fertilizer for farm fields. And especially now, we know much more because Maine has been very proactive about this. There are a lot of farms in Maine that have now been contaminated with PFAS that can no longer farm because they had had decades of biosolids spread on their fields as fertilizer. And so that has that's become a very big issue in the agricultural community. So anyway, all of these things eventually then result in our the PFAS ending up going into our water, our soil, to our atmosphere, and sadly into being humans being exposed to it, mostly in specific areas to drinking water. But for people who are not exposed to drinking water, the next most common exposure is actually through fish. There's also things like food wrapping. So I tell my kids and my students, don't be eating those microwave popcorn bag, popcorn, because that liner in those bags is also PFAS. So something that you would never think. So what happens once it gets out of these sources? What we know is that, from overland flow and runoff, we end up having the PFAS go into surface water bodies. And, these are, and also, as I said before, private drinking water, plant uptake from areas that had biosolids spread, and the wastewater treatment facilities, which are concentrating all of the PFAS from all of our residential use of PFAS. Eventually gets into these water bodies, and that's where we come in as aquatic ecologists. We have been, wondering what kinds of PFAS are getting into what kinds of aquatic organisms. We studied mercury for a long time, and we know that mercury was bioaccumulated and biomagnified. And others have been studying those questions around PFAS. And so questions like, well, are PFAS bioaccumulated in fish and in vertebrates, as you can see on the bottom there? Are they accumulated more in things that feed in the benthic environment, the sediments, or are they higher in ones that are feeding in the water column? Are they highest in top predator species like mercury is? So there are a lot of questions around PFAS that are being answered right now. And we just don't know nearly as much about PFAS as we do about methylmercury. So I'm just move to one of our studies that came out a couple of years ago. My former technician led this paper, and basically it was a review paper to look in the literature to see how many studies are out there. This was in like 2021. How many studies are out there studying shellfish, both bivalves like mussels, oysters, clams, and cephalopods like squid and octopus, crustaceans like lobsters and shrimp, and gastropods like snails and conch and things like that. And what you can see is these were the studies at that time, which isn't a ton of studies, actually. You can see the colors represent when they were done. So the pink are earlier, 2005 to 2009. The yellow is later, and then the blue were the more recent studies. They're from all around the world. A lot of them are from places where shellfish are harvested, like the Mediterranean and in the North Atlantic. China has a lot of, harvesting of shellfish as well. They also happen to have a lot of sources, as do we. And so that study basically was trying to look at different kinds of shellfish. So if we look at how shellfish obtain their PFAS, you can see that they are taken up by both bioaccumulation, which is A, and B is bioconcentration. And bioaccumulation is taking it up through your food and through your water combined. B, bioconcentration, is just taking it up from water. And then C is biomagnify, which, if you haven't come across that term before, it just means that with each level of the food chain, it increases in concentration because those animals are getting it from their food, and then they assimilate that into their tissues and pass all of that contaminant on to the next higher trophic level. And so you can see that things like snails, clams, mussels, and oysters are really what we call them primary consumers. They eat mostly plants and broken down material that's in the mud. And then more predatory organisms that are shellfish are like shrimp, squid, crabs, and lobsters. And so we wondered whether what were the patterns? So this next figure is a bit much, and I'm not really wanting you to process all of it, but it is basically looking at averages of the alphabet soup of all these different compounds, and going from top down to the bottom is going from shorter to longer chain. And what you can see is really in brackish water, freshwater, and saltwater, there are different compounds that are present and have been measured. And also these are the sulfonates on this side, and these are the carboxylates on the left side for bivalves, so clams, mussels, oysters, and then for crustaceans like shrimp and lobsters. And what you can see, I just listed here, the ones that tend to be higher in concentration in these. For the bivalves, we have a lot of PFOS. PFOSA, which is not one that I mentioned earlier, but we're seeing PFOSA as well in crustaceans and lobsters. But PFOS and PFOSA are ones to look out for in those kinds of marine organisms. Now, all of this, some of this, this is a review paper. So some of these things have been studied in marine organisms for a while, but there is really not that much data in general in marine ecosystems. So the interesting thing that then we did with these data from this review was to then take the data from the review, use data for level of seafood consumption or shellfish consumption, and calculate whether any of these compounds actually present risk at the 50th percentile consumer. In other words, average consumers of seafood, of shellfish, and high seafood consumers, or 90th percentile consumers. So if we look at the average consumers, and then we look at this gray line, what you can see is that this gray line is the line for what is called a hazard quotient. So this line is at a level of 1. And anything that is above that suggests that there is potentially a hazard from consuming those shellfish for that particular age group. So for adults, there is some potential hazard from consuming shellfish at the 50th percentile level for PFOS. Also for women of childbearing age for PFOS, because the circles represent PFOS, and as well as for children. So the PFOS that is in shellfish in the levels that were measured in that review can present potentially hazards to for PFOS exposure. In the 90th percentile, so the people who eat a lot of shellfish, you can see actually more of the compounds could present risk, including PFOA, PFNA, and also PFOS. So we're getting some indication that at levels of PFAS concentration in the environment and organisms that have been measured, that there could be some risk of exposure to people who eat shellfish in this case. Okay, so I'm going to move now to another study that we did and are still writing up, but we also had this great sample set of a whole bunch of demersal fish species. So demersal fish are fish that feed in deeper waters and near the sediments. And our question was, what is the level of bioaccumulation of PFAS in these fish species from the Gulf of Maine? We were able to get a whole bunch of fish from the National Marine Fisheries Service that does fish surveys of these particular species of fish, including silver hake, red hake, spiny dogfish, skate herring, and Atlantic cod. They do surveys every 6 months, and we were able to submit a request for them to provide tissues to us. So we didn't have to go out and get them. And then we hired a Portsmouth fisherman, David Gaithel, to go out and get us these inshore fish from some of the same species. So you can see with spiny dogfish, we have ones closer to shore, a little farther from shore. For skate, we have ones that were closer to shore and ones that were farther from shore, because we wanted to see whether there was any difference in distance from, human influence. So we then, here's a picture, a graph of the different species, as well as the screening levels for New Hampshire adults and New Hampshire children. And what you can see, so this is a lot of colors and a lot of a lot of compounds. And like I said, this alphabet soup of compounds is important, and when we're studying it, and it can be very complex to explain. But let's just say, if we look at PFOS, which is, as you know, one of the ones we are seeing across some shellfish species, and we know that it's being going to be regulated by the EPA. The inshore bar is this one, and the darker red, the offshore, is the red. For winter and little skate, you can see that the offshore individuals had lower concentrations than the ones closer to shore, which kind of, suggests that there are some land-based sources that are probably contaminating them. And the other thing you can see is that the inshore skates and are actually above the screening level for children in New Hampshire. We can also see that for some of the other, for red hake as well, you can see the PFOS inshore is higher than the PFOS offshore, and same for Atlantic cod. So we do, across some of these species, not all, see this inshore higher concentrations than offshore. And I should say this is Nate Gifford, and he's our former technician, but he's leading this paper, and so that's who did all the data analysis and certainly did a lot of the processing of all of the samples. But if we look at another way of looking at the data, is the percent composition of each of the compounds. So what you can see here is that for Atlantic cod, in the first one, inshore, you can see that the dominant color in this bar are PFUA and PFTRDA. And those two compounds are 11, PFUA is 11 carbons, and PFTRDA, the blue, is 13 carbons. Now, those two compounds, which are high in these fish, and in every one of these species, those two compounds appear to be the highest in the proportion of total PFAS. Those are actually compounds that are not regulated by anybody, and we don't have any screening levels for them. And they are higher than PFOS, PFOS, which is the orange, which you can see in these marine fish are really a small percentage of the total. That is completely different from freshwater fish. Freshwater fish are much higher in terms of percent composition in PFOS. So there appears to be a difference between marine fish and freshwater fish. And because freshwater and freshwater fish have been studied much more than marine environments, PFOS has been a real focus of everybody's attention in terms of establishing screening levels and measuring it in drinking water. We know much less about these other compounds that now we're seeing are high proportion in marine fish. So that's one of the findings of this paper, that we think is important because we really need to keep moving forward and studying some of the compounds that we're actually being exposed to more. And most people, people do eat freshwater fish. Of course, all of you recreational fisher people eat freshwater fish, but most of our fish, most people eat marine fish. So it's important to really understand what is in marine fish. The other, this is, let me just see what time it is. Okay. This is just to show, and I'll just tell you what this says. So I just mentioned that the PFUDA and the PFTRDA were dominant in the fish, and we measure something called stable isotopes. So in this case, we measured C13, which measures this carbon source of the organisms, and N15 measures the trophic level. In other words, the higher the N15, so to the right side of the graph, the higher the trophic level of that fish. So what we're trying to understand is, do these compounds relate to the trophic level of the fish, or are they related to where they get their carbon? And in the case of C13, the farther to the left, the more negative the values are, the more that carbon, their food source, comes from the water column. And the more to the right it is, the more their food source is coming from the benthic environment. So what this is just showing you is that for both these compounds, that in fact, the PFTR, these two particular compounds are higher in fish that are feeding in the mud. So benthic feeding fish that eat organisms that live on the surface of the mud or in the mud, are actually getting higher amounts of these two compounds. There's a slight relationship with trophic level, but it's still pretty weak for PFUDA. So that's what we've sort of found in that demersal fish database. I'm going to now move to another study that we did, and this one is by my lab manager, Christine Gardner. And she is our PFAS expert in our lab. She has her master's degree in PFAS. And what this was, we wanted to look at the shellfish around the different locations in Great Bay, New Hampshire. So for those of you who don't know, this is the former Pease Air Force Base in Great Bay. And the former Pease Air Force Base used a lot of aqueous firefighting foams. And as a result of that, the groundwater in these communities around Pease are contaminated with PFAS. And we went out to ask the question of whether these shellfish that are found, because these are all areas where people go recreationally shellfishing, wanted to find out if we would see a relationship between how close these shellfish beds were to Pease, the former Pease Air Force Base. And we looked at soft shell clams, we looked at blue mussels, European oysters, American oysters, and razor clams. Oh, and I guess one thing I forgot to mention, right, was that the data for the fish was that the skates were high in PFAS. And in the case of these shellfish, what we see, if you look at this on the right, we have one particular kind of shellfish that is above the New Hampshire adult child and EPA screening levels, and that is razor clams for some reason. And we, at this point in time, do not know why, but the razor clams are extremely high. So skates have high concentrations in PFAS and razor clams have high concentrations. But again, looking at the percent composition, what we do see is that we do see PFOS, the brown, is dominant in razor clams, but the higher the TRDAs and the UDAs are actually quite prevalent in the soft shell clams, oysters, and blue mussels. So something about razor clams in terms of their composition, it could be that PFOS is just, concentrating much higher than these other longer chained are concentrating in these other shellfish. But the other thing to notice is this is the composition of the animals. look at the composition of compounds of the water and sediment. They look nothing like the bivalves that are living in the water and the sediment. So, but PFOS is still very prominent in every kind of shellfish. The biota are really dominated by these 13 and 14 chain compounds, and the water's dominated by these shorter chain compounds, the C4 and C8, which are these yellow ones down here and peach colored ones. So for us, it's important to look at what compounds are in which matrix, because we're wondering, if the concentrations in those water and sediment actually predict the amount that's in the environment. I should say, for these shellfish, we did not find high concentrations in anything but the razor clams, even though the sites, some of them are very close to the former Pease Air Force Base. So that's good news. And the water and sediment concentrations are very, very low as well. So our findings to date then, well, let's see. I won't go, I'll not repeat those. I just want to mention this study that we've just published. This is a study on seafood consumption among New Hampshire residents and looking at their seafood consumption using seafood consumption survey in New Hampshire, and then doing a market basket study, looking at going to a store, buying seafood, and seeing what's in the seafood that's commonly eaten. And so what you can see here is this is the percentages of people in New Hampshire that eat these kinds of seafood. So shrimp is very dominant in people's diets, as are haddock and salmon, canned tuna, lobsters up there too, scallops, cod. And so these are the most commonly consumed. And what you can see is the light blue is whether they're self-caught or not. But as you can see, for most New Hampshire residents, and this is also nationally as well, most of the seafood we eat is purchased as opposed to self-caught. A small fraction is self-caught. But that was one of the questions on the seafood survey. So this is to show, okay, tissue concentrations in shrimp. So we went and bought shrimp and salmon and haddock and lobster. And what we found was that there were some higher concentrations in shrimp, particularly of PFOS. You can see here, we've been talking a lot about PFOS, and in lobster. But here you can see, as I mentioned before, the PFUNDA, the 11 and the 13 carbon chain PFAS are higher in lobster, as is the PFTEDA, which I think is like 10 or 11. No, not 11, 12. It's maybe 12. So we took the concentrations from the market basket study tissues and did that same kind of calculation of risk for New Hampshire 50th percentile consumers and New Hampshire 95th percentile consumers, like the one like that calculation we did before for hazard quotients. So, but this is specifically for New Hampshire. And what you can see is for 50th, so average seafood consumers in New Hampshire do not have any real risk of exposure to PFAS from shrimp and lobster. But when we look at the highest seafood consumers, you can see that for PFOS, there is some potential risk for consuming shrimp for PFOS. And then for PFUNDA, there is some risk for consumption of lobsters. Now, I mentioned before that PFTRDA, some of these compounds, there are no screening levels. This screening level for PFOS is for New Hampshire. This screening level for PFUNDA is actually from New Jersey. So we don't really have a lot of screening level values for, the even 40 compounds that we already measure for PFAS. So anyway, so that's how we then came to this conclusion that we would, there may be some risk from consuming those two kinds of shellfish. You might have, this story was on NHPR recently, so you can listen to that one if you want to hear more about it. I think I'm going to stop there. I had a bunch of slides just to tell you that, well, I'll show you a picture anyway. We are doing PFAS fate in Great Bay food webs, which includes all of those organisms that are pictures on the bottom there. We already did our field work for that, and we are measuring PFAS in these lower trophic levels. In Great Bay, you can see the map up above. We also are studying 5 lakes in southern New Hampshire that already the state has identified as having high PFAS in their fish. And we also are going back this summer to a bunch of tributaries of Lake Champlain. So we're doing freshwater work as well as marine work these days. And I guess I should acknowledge certainly all my amazing collaborators and certainly also all of our funders. So anyway, if you have any questions, please go ahead and ask them, and I will unshare my screen. So, any questions?
We did have one question that came in the chat, and that is, have there been any studies on species that were raised in aquaculture farm environments?
There have been, I believe that, but not very much, I should say. And so we were wondering, especially because there is shellfish being farmed in Great Bay. But once we saw the shellfish data for the, for the other wild caught individuals, I think we're less worried now about the farmed shellfish in Great Bay. But in general, so farmed shellfish eat from, natural food webs, right? Because they basically put them out there in the bay. Farmed fish are often fed other sources of food, fish meal and all kinds of other stuff. So their exposure would be different than organisms feeding in the wild. And I actually don't know that I've seen anything on farmed fish at this point in time, but it's probably out there. The literature coming out on PFAS is like out of a fire hose. There are so many papers being published. You can hardly keep up. Yeah.
Are there any more questions that people would like to ask of Celia while we have her here? I'm watching the chat, watching for raised hands.
Well, I have one. There's one that just popped up into the chat too. It looks like Jay is asking, in drinking water, will PFAS be reported soon? Is that something?
So in New Hampshire, if you all live in New Hampshire, you can actually get your water tested and paid for by the state of New Hampshire. You should go to the New Hampshire DES website. That's been a really amazing thing that New Hampshire did. The legislature actually put a lot of money aside for drinking water testing in New Hampshire. And so, yes, for public systems like, Manchester, if any of you live down there, the Manchester water supply, I think, has already been, so public water systems have been tested. But now now that MCL that I mentioned, that US EPA just established, really made the maximum allowable concentration level way lower than what New Hampshire had before. So it means that water systems that were okay under New Hampshire's regulation will now, under the federal regulation, potentially be considered contaminated with PFAS. So I think, that MCL came out, and I think a lot of state agencies are really, going to be grappling with this. But if I were a New Hampshire resident, which I am not, I certainly would want to I did actually have I live in Vermont, and I had my water tested because I was part of the US Geological Survey test to get my because PFAS costs about $500 a sample to get it measured. So, hey, if the state of New Hampshire is willing to help you pay for that, I would go, because you don't know. I mean, there are known contaminated systems, water systems. Like I said, the people who live around Peas know that their wells are contaminated. But if you live next to farm fields that have been, using biosolids, your groundwater could be contaminated too. And in New Hampshire, we have so many, and Vermont too, we have a lot of private wells. So your local, town is not going to be measuring PFAS in your private well. So I would encourage you to go get your water tested. That's what I would do if I was in New Hampshire. And if you live in Vermont, then you're going to have to go pay for it yourself, because Vermont doesn't have that. Sorry. Yeah.
Let's see, a couple other questions.
I see Amy, my dear friend and classmate, Amy Simon Berg, asked a question.
Yeah.
Yeah. So Amy, if you find your personal waters contaminated, where do you go to remediate? So there's a whole industry now that is emerging that is for remediating drinking water. It's expensive, and it has to, and it's a lot of activated carbon filtration, and some of those towns, like in the Peas area, those municipalities have had to spend a lot of money on getting their municipal water filtered. Yeah, so there are technologies for filtering it, but then, of course, then what do you do with the filter? There's just all kinds of disposal issues, but you can have it removed from your drinking water. Yeah.
And Nina asked a question about PFAS moving through underground aquifers. Is that as fast? I think the question is it as fast as it would be in surface waters?
Good question. I'm not sure. I would doubt that it's as fast, because, the velocity of like a river or stream is much greater than I would think movement in groundwater is. The only problem there is once it's in the groundwater, it's virtually impossible to remove. And in fact, I'm funded by the Department of Defense, and they have all these sites, and they are literally funding people to research ways of destroying it in groundwater, literally putting these gigantic heating probes into the ground to heat up the ground to try to destroy it. But it takes very high temperatures to destroy these compounds. So yeah, so I don't think it moves around as much, but it just, once it's there, it doesn't get flushed out like a river or a stream does. Yeah.
Mm-hmm. And Bruce was asking if you have a feeling as to whether it's a greater concern in drinking water or in fish food, like consumption through fish or consumption through drinking water.
Yeah. So for people who are not on contaminated well water, the dominant exposure route is seafood consumption, fish consumption. And in fact, there was a European food safety study that was done, and it was something like 80-something% of people's exposure outside of having a contaminated drinking water system was from fish consumption or seafood consumption. Yeah, so that's why we are being funded to do these studies, because it is an exposure route. Yeah, sadly.
And Elizabeth, you had a question?
Yeah, I was curious. You mentioned that you had some advice that you gave to your kids not to eat microwave popcorn. And I wonder, with all of the research that you've done, what are some of the changes that you've made in your habits, or that you recommend to others to reduce
Yeah, great question. I think it's still a struggle. So I've learned this. I know that my ski wax for my cross-country skis is PFAS. So I went, and my husband bought some non-PFAS ski wax. So ski waxes, boat waxes, floor waxes, believe it or not, have PFAS in them. In fact, I learned recently that some of the schools up in Maine, they contaminated their groundwater around the school because when they were waxing the floors, they dumped the wastewater down the drain that went into the wastewater. So waxes And in fact, the international ski racing, federations have already banned the PFAS waxes. So I know I have this block of PFAS wax in my ski bag, not sure what to do with it. So I don't have advice about what to do with these things. I also know that when I bought my car a couple of years ago, I sprayed it with Scotch Guard because it has beige upholstery. And Scotch Guard is like PFAS, and I'm sure I was inhaling it for days. It's in so many things that you don't even realize. I mean, I have a Gore-Tex anorak. What am I going to do with that thing? I think that what we have to do is to get it out of our products. So the states in New England, Maine, New Hampshire, Vermont all have product bans that are being discussed in the legislature. And Maine is farther along and trying to ban products. And New Hampshire and Vermont are, involved in those kinds of things. I really think that we have to ban the products so that they are no longer purchased, so that these companies don't produce them. I mean, it's that. I don't know what else. So it is that we have to stop it at the source, which are, both 3M and DuPont have been really are in enormous suits that are class out, bunches of states. But I mean, the problem is that we have a whack-a-mole problem, is that when the PFOS was discovered and PFOA were found to be associated with human health effects, they did the responsible thing. They stopped manufacturing them, but they also then produced Gen X. And Gen X is now considered, a human health hazard. So fortunately, we don't have very much Gen X up here. It was manufactured down in North Carolina. So one big river system, Cape Fear River, has tons of Gen X in it. So I think we have to be more educated consumers. And so I'm trying to educate myself as well, but I have certainly learned that a lot of the products, even in my house, I've actually worried because we have trouble with getting our samples from the field contaminated. And I realizing that it's in floor wax, it could be in our lab, right? I mean, so there's just all kinds of things like that. But I do think that we have to, there are many in these product bans, there are many products for which there is no replacement. And so I think that those probably will not be able to be banned. A lot of medical equipment has PFAS in it. So, and these aqueous firefighting foams, there is not an effective replacement yet for those. And those are, huge safety, concerns with fire. So it's complicated. I would say for me, I love getting the chance to talk to you all because I think I've learned so much about PFAS. And I think as we just need to be educated consumers, I'm trying to figure out what I shouldn't buy and what I should be involved in. There's just a lot going on around PFAS right now, but definitely no microwave popcorn.
Karen Ebel put a note about bills in the house. Karen, would you be willing to unmute and speak about that?
Yes, Karen. Go, Karen.
Yeah,
That's
sure.
great.
Hi, thanks. So I'm Karen Ebel. I'm a state representative in New Hampshire. I represent New London and Newbury.
Wonderful.
And as one of the ways to try to stop all the PFAS use, it's exactly as what your speaker said. It's you got to stop using them in the products originally. And so I do have a bill that passed the New Hampshire House, actually 233 to 140. I was very excited. And we just had our Senate hearing. The products that I'm seeking to ban is carpets, because that has a huge amount of PFAS in it. Water, things like Scotch Guard, cosmetics, believe it or not, if they stick to your face and you know, if you're running and they don't drip, it's probably because it has PFAS in it. So cosmetics, juvenile products, because many juvenile products for kids 12 and under have PFAS in them, food packaging, textile articles in the home like draperies and things like that. And I think that's it. I believe it or not, dental floss frequently has PFAS in it. I haven't included that. I tried to be more limited. Ski wax, as she said. Some states are banning apparel. Some states are banning cleaning products. There's a myriad number of products out there, and I actually have excluded medical devices from my bill just because it is complicated. There aren't replacements for it. New Hampshire was in the vanguard, believe it or not, in banning PFAS added firefighting foam back in 2020. And many other states have followed suit. If anyone's interested, the bill number is HB 1649. The committee is Energy and Natural Resources in the Senate. They're going to be taking a vote on whether to recommend passage on Tuesday. I'm anxious about it, to say the least. I was really appreciative that New Hampshire Lakes, a lot of environmental organizations supported it, but also landfill operators, because they're dealing with it, wastewater treatment plant operators, drinking water. I got everybody from New Hampshire Lakes to these big landfill operators. We're all trying to ban it. So I'm hopeful that we can follow in the footsteps of Maine and Vermont. Massachusetts has a bill pending. And at least regionally, maybe we can do something with these things. So thanks for letting me talk about it.
Thank
Karen,
you. I
thank
think that was
you.
really valuable.
Yeah, that is really great to hear. Thank you for the good work, Karen. That is really
Oh, well,
wonderful.
you're welcome. You know, I got to tell you, all this, the alphabet soup you were talking about, that's sort of where a lot of people are snagged right now, because
Yeah.
it's how to define PFAS. And I'm trying to stick with the broadest definition I can so that you look ahead and try to capture new developments going forward. So we'll see where it all ends up. But yeah, definitely right in the middle of a lot of PFAS activity with this bill.
Yeah, well, it's certainly relevant these days. Oh, and the other one you mentioned, dental floss, Glide. Glide dental floss is definitely.
Don't buy
I used
Glide.
to use Glide all the time. I don't use that anymore. So yeah, but I think we have to, and also, you know, for a lot of things, even in my lab where we don't even know, you call a manufacturer, they don't know if there's PFAS in it, because we're trying to make sure we don't contaminate our samples. And it's really hard to find out whether PFAS is in a product, in a, you know, an instrument, in a anything, in a pipette. We found out that pipettes sometimes have them in them, you know, so anyway. It's a wicked problem. It is a wicked environmental problem.
Well, thank you so much, everybody, for attending tonight. Thank you, Celia, for sharing your time and talent with us and all this great information. The recording will be posted on our website.